I realized that this is a really very complex topic, particularly for a non-zooplanktologist. I took here this picture, the scheme from Benny Hansen from a review article. And copypods are diverse in terms of their strategies and their life cycles. So not every copypod species does the same. As Ida explained also in her talk, they have these two types of eggs, these subcutaneous eggs for normal growth at good conditions that they can continuously hatch when they sink down to the sediment or even before. And then these diapause eggs that are assumed to be produced at unfavorable conditions, for example, in late autumn in a system. There's also temperature could be a trigger, but others, of course, the light conditions, the light cycle, and genetic factors. And these diapause eggs, they sink to the sediment. They constitute a kind of seed bank in the sediment, which is very interesting. They can stay there for prolonged times. I think in freshwater systems, diapause eggs of more than 300 years have been found that are still able to hatch. And this phase we call refractory phase, that they need a certain refractory phase before they hatch these diapause eggs. Well, and then it's getting more complicated because these subcutaneous eggs that fell on the sediment become quiescent eggs when the conditions are unfavorable. And unfavorable means, for example, you have hypoxic conditions above the sediment, or you have some sediment resuspension. They get covered by sediment. That means they do not hatch, but that could be delayed until, again, the conditions change and they hatch later. And one important thing here for the diapause, one eggs deeper in the sediment, they conserve the genetic information of the time when they were produced. And imagine that we have microevolutionary changes within the copypod population, but later on, for some reason, these old eggs might hatch and they carry the genetic information from the past. That means it's like a conservative factor, a little bit dampening microevolutionary developments in the population, possibly. Well, and then temperature and hatching here for these subcutaneous eggs that become quiescent eggs. Temperature could be a trigger, among others. For the diapause eggs, temperature could be a trigger. So we have everywhere, we have temperature as one important factor. So this relates to the overall topic of warming and increasing water temperatures. So we should assume that really temperature should impact the life cycle of the copypods in various ways, the production of diapause eggs, the hatching of these different eggs also. And so it's worth to go deeper into this topic because something should happen there. And of course, there's more than only the temperature. You have also advection. This is a scheme for the succession of, well, from one of my colleagues, made it for the German Baltic coast for the succession of marine and estuarine copypods. Advection of water masses plays a role, but also the hatching from the egg bank, the dormant egg production, that's in blue here for marine copypods, and in orange for estuarine zooplankton in Greifswald Bay, so a bay close to our institute. And then you have these here, the hypoxia at the bottom border, which influences the hatching success, and you have the sediment resuspension, which also influences the hatching. And he also said that this is still poorly understood how this affects the life cycle and reproduction of the copypods. So one of the big questions also raised in the thesis is, can we see adaptive modifications in the life history in response to environmental changes? And that is something also we can discuss later, I think. Well, and then also biofilms. Biofilms are so complex, really a high complex structure, and that's the next topic Ida approached in her thesis. And these biofilms are consortia of really different organisms, but when you look in typical review articles like this one here, they show only bacteria. Of course, microbiologists were mainly interested in bacteria, and there are so many fascinating things going on in the biofilms where the bacteria are building up a matrix, an exopolysaccharide matrix, which serves many different functions, protection, for example, but also enables communication. There are many communication systems between bacteria have been found in biofilms. And then we shouldn't forget that there's always an ongoing succession of biofilms on natural surfaces. So bacteria attach to a biofilm, then they are building up this exopolysaccharide matrix. The biofilm grows and forms a complex three-dimensional structure, but later on it might detach again due to current, due to larger crazers and other factors. So we are always have to be aware that there is an ongoing succession. And then when you look at real biofilms in nature, by different types of methods, scanning electron microscopy or confocal laser scanning microscopy and staining with fluorophores, you realize that the real biofilm do not consist only of bacteria, but here you have diatoms, you have many different types of algae, you have heterotrophic protists there. So it's a much more complex community than what they showed in the textbook examples normally for biofilms. And this is exactly then what Ida approached in her thesis, and by covering both the microeukaryotes and the prokaryotes in the biofilm and asking these important questions about which we currently really do not understand a lot. And I am not really aware of previous studies that have investigated the warming on the biofilm, natural marine biofilms. Okay, well, just to sum up, Ida has applied a wide range of techniques and approaches in her thesis: sediment and biofilm sampling, nucleic acid techniques, and all what belongs to that, the assessment of zooplankton, so really going to very different trophic levels, hatching experiments with zooplankton, bioinformatic and statistical analysis, and then these translocation experiments, these really with a very complex experimental design and a lot of work. And I think she has achieved for these two topics, or for this topic in general, many new insights that we now better understand how these are two understudied components of the coastal Baltic Sea might respond in a changing future climate. At least there are still many open questions, of course, because the topic is complex, but at least some of the gaps, I think, have been filled with the thesis. Thanks. That's all for the moment. Oops.
Thank you very much, Klaus. We will now have a break. All right, ladies and gentlemen, have a seat. It's time for the discussion between the opponent and the respondent. So you may ask all the questions you would like, Klaus. And Ida, you answer what you...
Microphone. Is microphone switched on? Yes.
It sounds beautiful. So I give the word to you.
Okay, let's start, Ida. Maybe let's start with some general things about climate change and warming. What was your personal experience? I mean, before you started the thesis, were you already concerned with climate change? And did you know about, I mean, more than we read in the media, I mean, did you know about what happened with climate change in the Baltic Sea already?
I did my master's thesis about climate change.
Very good. And in which direction was that?
It was more so I went to Oslo and I did more like how did the current climate change affect like speciation, extinction, more like biogeography in relation to climate change, because there have always been climate change, like since dinosaur time, and climate change and Earth have changing, but the problem now is that they are anthropogenic caused and they are much faster than any other climate extinction. So you have these big 5 extinctions, and this one, there is a background extinction rate, but the extinction right now is faster due to the anthropogenic caused climate change.
That's right. And I didn't get, but on which organisms did you work on in your master thesis? That was
not specific organism. It was like general
processes.
It was not specific some organisms.
But on which system? That was on which system? What? On which system? On which aquatic system?
I only had 1 semester, so I couldn't really do climate change experiments.
Yeah.
So, it became just literature I was using.
Okay. So just for my curiosity also, I mean, in your PhD thesis, you deal with microorganisms, with zooplankton, with sediment, with plankton. In which of these fields did you have some experience from your previous work? Because it's unusual to cover so many different topics, and most people, well, they study microbes and then they only study microbes, or they make zooplankton or sediment. But were you already familiar with one of the fields before?
Well, I had microbiology before, so I was aware of like they make a biofilm, become anaerobic inside, and blah blah blah. I already knew that, but I have never worked with copepods before.
So you were more familiar with the microbes than with the copepods.
But also,
Okay, and the Baltic Sea, I mean, you have experience now from benthic and pelagic components. And well, I'm wondering, I mean, both are affected by warming in the future, obviously. But what is your opinion on the differences, or which of these subsystems is more affected or more faster affected than the other, the pelagic system or the benthic systems? Do you see differences there? How they respond to climate change, or how they also maybe adapt to climate change?
Yeah, so if you look at the benthic, of course, it's more affecting the microbes in the sediment because it's directly touching the sediment, and also the biofilm is on stones that is laying on the sediment, so it's affecting more what I'm working with, but the copepod is in the entire water column. So.
But what do you think? Can we say that benthic sediment systems, there will be a delay how they are when they are in their response to warming, or are they less affected than pelagic system, plankton systems in general?
Well, plankton, they can just migrate. The microbes cannot really migrate as fast. So.
Yeah. But okay, you think of higher trophic levels. Huh?
What?
Are you thinking of higher trophic levels that they migrate, or they respond differently to warming?
I'm not really sure I really understand what you are asking.
Yeah. Well, we continue with the Baltic Sea. No? Do you think the Baltic Sea is special, no? in many aspects, different? Which aspects of the Baltic Sea were important for your study? What do you think? What speciality of the Baltic Sea?
So the Baltic Sea is really affected because it's like a closed area. It's not like the open ocean in that way. So with it, it's kind of stays more. There's a lesser turnover of the water because it's not like an open ocean. It's more like a closed system.
And what are the implications of that? That it's
There could be it could be more like the climate hazards could be more severe in such a system.
Yeah. And what do you think about the salinity? Does salinity play a role for your communities that you have studied?
Of course, salinity is playing a role. We haven't looked directly on it, but with climate change, there is evaporation. As we see in the Mediterranean, there is evaporation of water, which increases the salinity there, and then organisms will have to either adapt, move, or they will die. It's how it is.
But also you can have the reverse effect in the Baltic Sea, that you have a dilution, no, of salinity by the rivers.
Yeah, if there is fresh water coming into, then there is a dilution.
Yeah, but then the question is, the communities you have studied in the sediment, biofilm, zooplankton, are these communities marine communities? Would you call them marine communities?
Brackish kind of communities.
So typical brackish communities, you think? Yeah. Okay, and how would you consider the importance of the sediments for the overall ecology and biogeochemistry of the Baltic Sea? If you say what is how important are the sediments? Are they particularly? I mean, I know sediments are important everywhere, but why are they so important in the Baltic Sea? What do you think?
There is a lot of geochemical cycling in the sediment. We see have carbon, nitrogen, sulfur, methane is happening in the sediments. So if something changes there, you change the environment.
So probably more is happening in the sediments than in the water column.
Yeah.
And are there other, I mean, I mentioned a few other features and problems of the Baltic Sea. Are some of those, are there other factors that are relevant for your study besides warming?
Well, we are studying a natural system, so of course we have natural fluctuation, not only the warming. We have everything that is in there in nature because we went out in nature and we studied this. If we had taken it to the lab, we will have to simplify and we wouldn't capture as much as we do in a natural environment.
And these problems, eutrophication and oxygen depletion, are these factors? Were these factors relevant for you, for your systems that you studied, for the two bays also?
Yeah, there is a lesser oxygen with the heating, and that could also be a problem for organisms.
Ah, yeah, okay. And I think you didn't mention oxygen. So how strong was this oxygen depletion in the heated bay? I mean, to what level was oxygen declining there? Do you remember?
I don't remember the exact numbers, but it was lesser oxygen in the heated bay than in the two other environments.
But no hypoxia, no real hypoxia, no? Okay. Means that really not very low levels, no, of oxygen.
I don't remember the numbers.
Yeah. And well, I mentioned that the bays, well, they are a nice natural system. That one has been heated, the other not, but that there could be other environmental factors that are different between the two bays and that have an influence on all these studies, not only yours, but also the previous PhD theses that have been performed. What other factors could be different between the two bays, to keep this in mind, both in the water and in the sediment? What do you know some of these features of the system that are slightly different between the two bays?
Well,
not everything is similar, I guess. No?
There is another sedimentation rate in the two bays.
Sedimentation rate?
Yeah, it differs, and there's because we were not there 50 years ago to take samples. But we just assume, because there's only 1.5 kilometers in between them, that they are fairly similar. But it could be that from the beginning, before the warming even started, that there was differences between the bays. But we don't know because we were not there 50 years ago.
Yeah. And well, you already mentioned oxygen, but oxygen that's related to temperature maybe is different. And what about nutrients?
Yeah.
So stimulating, there are also some differences in nutrients regimes, or?
Yeah, there's differences in the nutrients. We took nitrogen, phosphate, and sorry, one more.
Silicate maybe.
Yeah, exactly.
And so that's where is it higher? Where are nutrients higher?
Oh, I think it was the heated baby.
Yeah,
Higher
and that means it also has higher primary production probably,
Yeah.
and you mentioned higher sedimentation rate.
And also in the heating, there's a higher turnover of carbon.
Yeah. Yeah, you see. But all these factors, I'm wondering when you compare, let's say, biofilms or whatever, some communities you compare between the two systems, and then you say, well, temperature has an effect. So it's the effect of temperature. There's a change in the diversity. But when so many other factors are also changing, maybe a little bit related to temperature, but also nutrients productivity. So is it still then possible to say, well, temperature is a reason why we have a changing diversity? Couldn't it be also that it's higher productivity?
Yeah, it could. Is also what I said and why we took the wave exposure into consideration, because the tube bay, when you go there, it's just like harmony and peaceful and nice. When you go to the gradient, we got kicked over by the waves. They are very, very different in many different ways, and also the turnover of nutrients is higher the more wave activity you have. So there are differences between the bay, and also when we look at the community composition, you could see it didn't cluster like tick, 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 like the gradient clustered a little bit away, meaning that there is other factors than the temperature only that is affecting the community composition in the tree environment.
So one has also to consider all the other environmental factors that are shaping the communities. You mentioned this gradient. I mean, so you mentioned the gradient, the gradient.
Gradient, yeah.
For me, it was hard to understand how this gradient is formed and maintained somehow because you mentioned there's wave action, there are currents, when you have higher wind and waves and action, and then there's a lot of mixing of water masses going on, I can imagine, in the open, because it's open. How can you have a gradient? How is it maintained physically, the gradient, despite all these mixing?
It's a gradient of the temperature, but of course we also acknowledge that there are other factors that differs between the gradient and the tube base.
Yeah, sure. But why is this gradient there? Is it always there? Is it always a gradient, or is it sometimes disturbed and disrupted? And because of this mixing when you have mixing of water, normally you have no more gradient, I would think.
Then if you look at the temperature graph that I show, you can see there is a gradient. So it is warmer towards the heated bay, and then the more, because so the heated bay, there is a constant flow of water. So it takes water from somewhere far away, it put it in, and there's constant flow, and the water always go like this all the time. So it's always moving warm water, and then the longer you go away from where the warm water starts to get out, the more cold it is, because there's a constant flow.
Yeah, I see. Maybe some fluctuations when you have a strong storm or wind or so, but in principle, it's a continuous flow. I understand. Okay, maybe if we discuss your first paper, the climate warming disrupts zooplankton phenology and overwintering strategy. First, maybe some general remarks about copepods in the Baltic Sea. What do we know about copepods in the Baltic Sea? Do we have also typical brackish water taxa? Do we have marine taxa? What kind of species do we have to expect in the Baltic Sea?
So acacia was for sure the most dominating.
Is this a brackish or a marine species? What do you think?
I think it's braggus.
Okay.
So there were different species also, but it was mainly acacia, and the other one was called Euromota.
Eurythemura or?
Do you want? I can find the name. It's in the supplementary. Yeah, acacia was there, and Cyclopo or Euromora was the tree that was really dominant.
And do you think in the Baltic Sea that we also find real marine taxa that are normally in the Atlantic or the North Sea and in the Atlantic? Do we find such taxa also in the Baltic Sea?
So they have traveled all the way from the North Sea?
Yeah.
Well, it could be.
Yeah, there are some. And brackish water species, no? We also have. Do we have other zooplankton in the Baltic Sea except copepods?
Yeah, we have also other zooplankton, but we focus on copepod.
Yeah.
In a future study, we could take into consideration more species, but since copepod is the most abundant, we look at the copepods.
At least in your area. Maybe it's different when you go to Finland in the north. Maybe you will find other groups of zooplankton that are more abundant, like cladocerans, for example, I was thinking of. Well, now you made it in the paper, you made a strong statement in your conclusion, saying that results seem to suggest that copepods adjusted their life history in elevated temperatures by relying less on the strategy of using sediment-stored dormant eggs and instead started their spring development earlier when phytoplankton food was available. So you suggest like a shift in the life history from less dormant eggs, more reproduction direct in the water.
Yeah.
This is an interesting conclusion, but I'm not the specialist on copepods. But I discussed this paper with my colleague in the institute, and he is a zooplanktologist, and he says he doesn't believe the conclusion. Okay. So let's discuss the findings, and maybe you can convince me about your conclusions and about your interpretation of the results. So what you showed is that in the warmer bay you have a faster development of copepods, also of phytoplankton, the chlorophyll, so they grow faster, which is simply an explanation because of increased temperature. Temperature promotes faster growth. This is not surprising. This is what we expect, that they grow faster, I would say. But then the interesting thing is the hatching experiments, that you find much more hatching from the control bay compared to the heated bay.
No, there were more hatchlings in the water column in the heated bay compared to the control bay.
But what do you mean with hatchlings in the water? What is that? Hatchlings in the water?
Napoli.
Napoli?
Napoli.
Napoli. Yeah. But I mean the experiment that you have more hatching from eggs in the sediment. You took the sediment, increase the temperature, then you observe the hatching of Napoli, and that was a higher in the control bay.
Yeah, meaning that there are more eggs. So we had sediment from both bay, and we put them, as I showed in the picture. And then the sediment that we took from the control bay, there was more egg that hatched.
Yes. Were there really more eggs in the sediments? Did you count the eggs in the sediment?
I spent more than a month trying to count eggs, and it didn't went well.
Yeah.
Because I was counting all kind of other stuff that I thought was eggs. So we really tried, and I had pictures, but I failed counting the eggs.
Okay. I never tried to count copepod eggs in the sediment. There's not a good method for that. It's not possible to stain them somehow, or So to better visualize them in the microscope, is it not possible?
Yeah, I was trying, but for my eyes, everything else also looked like copepod eggs, so it didn't really went well.
And I also wonder from the hatching derived from the eggs, but what kind of eggs were that? Were that these really dormant eggs, or were that quiescent eggs? You mentioned, you remember I showed that in my scheme, that a normal reproductive egg becomes quiescent, they say in the literature, when the conditions are not so good in the sediment. And now you do the hatching, and you do not really know from where the nauplia are coming. Do they come from real dormant eggs, or do they come from the quiescent eggs? I think we cannot distinguish, right?
If we should have known that, then I should have known how to look at the egg and know the difference, and I don't know.
Yeah, but it's not possible. I think nobody can do that. Nobody can distinguish these types of eggs. There's not what I understood, there's not an established method to differentiate these type of eggs. It just means when you have hatching, it could come from both types of eggs. Maybe it's more the timing. The timing is more important when you do the hatching experiments. And for example, when did you start? In which month did you start with your hatching experiments?
So we took the sediment just before the hatching would start. So to make sure, because they will put the eggs and then they will stay there until environmental cues like temperature and light. So we took it just before that started.
Yeah, but in which month was it?
I think it was March.
March.
Yeah.
Okay, so it was March. So it might have looked different if you took, or if you have taken the sediment course in November and December, maybe in this period, where they are producing diapause eggs, and then to see how many nauplia are hatching then. It might look different at that time of the year, I think, than later on. And one possible reason that we probably do not know is in the heated bay, you have quite high temperature. I saw that it's never below 10 degree, essentially. No. So that means you could have had hatching all the time before you started your experiment.
Yeah, in the heated bay.
In the heated bay. So they were hatching all the time, and at the end, when you start with your incubation experiment, there are not many more eggs left in the sediment because they were already hatching before.
So, yeah, okay, we should have taken the sediment earlier.
Earlier, no? Okay. For example, in November or December, then, and maybe then several times. And maybe in November, if we would have taken it in November, there would have been not a big difference because there were still so many eggs in the sediment of the heated bay. But now in March, they are already gone because they were hatching earlier. And that's why it's a discussion whether this is really an adaptation in the life cycle of the copepods, or whether this is due to the fact that you couldn't observe the hatching because it was already occurring before at higher temperature.
Yeah, I see. You should have taken the sediment course earlier in the heated bay at least.
Right, that would be something maybe for the future, for a future study to really look for these life cycle changes in the copepods in the system. It could be, but at the moment it's a little bit speculative, I would say, right? Yeah, and I mean, it's interesting with these if there are changes in the copepod population and the phenology of the copepods. And you have mentioned in your talk mismatch. So what kind of consequences can you imagine when the copepod population is changing in the system? Which other components of the food web are affected, and why are they affected from the copepods? For example, if you start with microbes, are microbes affected by copepods?
In a way, so the copepod is a very important node in the food web because fish that feed on them are used for human consumption. So if the copepod don't have the high population when this fish is hungry, you have a problem. Then they will go feed on something else, and then there's more problems. And also this copepod, they eat a lot of the phytoplankton in the water that we do not want to bloom up and be toxic and make the water all green. So the copepod are actually really important node. And if something happened in the life history, that or they will create a mismatch, then we can get problems both escalating up and down the food web.
So maybe problems for the fish population to
Of
develop.
course.
And have you observed such a mismatch, or do we have indications in your study system for mismatches that have occurred?
Well, it seems like both the main food source and them have an earlier abundance, so that's pretty good times. But we haven't looked at fish that eat them, so it's something that could be done in the future.
Yeah, it would be interesting to know something about the fish, because we had this in Greifswalder Bodden, in this bay I showed. That's also one hypothesis, that there is a mismatch between zooplankton and herring, because the herring stocks went down, and some scientists say, well, that's because of the warmer late winter temperatures and faster development of zooplankton, copepods mainly. And they develop too early and the herring come later, and when they come, the zooplankton peak is already over, and there's not enough food. I don't know whether this is true. Well, now we had, I think the last winter was pretty cold, and with very low temperatures. It should be different this year than the mismatch, right? And so, do you know anything about the fish production in the heated bay? Do you heard about that? No.
We haven't looked at fish. That's something that could be done in the future. But,
whether there's more or less fish production. And okay, that's up the food web. And if you're looking down the food web from the copepods, you said they eat phytoplankton. Okay, that's true. But how could heterotrophic microbes be affected by the copepods, like your micro eukaryotes and in the plankton? Don't talk about biofilm. In the plankton, bacteria, protists, flagellates, ciliates, are they affected by copepods?
Well,
what do you think?
If the copepods eat them, of course they are infected.
But whom exactly do they eat? Do they eat bacteria, or?
They eat the phytoplankton.
Yeah, and heterotrophic protists like ciliates or so.
I'm not sure if they eat them.
Yeah, they like them, I think.
Okay.
Because they move, they eat a lot of the ciliates, and then you have less ciliates, more flagellates, and that's how this cascades down to the bacterial level. But they cannot eat bacteria directly. They are too small. Too small for them. Okay, and the coupling? No? We are now in the pelagic system with the zooplankton. Now we will soon move to the benthic systems. But what do you know about the coupling between the benthic and the pelagic systems, the so-called bentopelagic coupling, which is often being discussed? What kind of mechanisms can you have that you couple, that you link the benthic system with the pelagic system, for example? By which organisms or by which processes are these two systems coupled?
So of course the copepod can be in the entire water column, but also geochemical cycling is of course affecting, because they put it into the water and it's affect the water was in the water.
The nutrients, you mean?
Yeah, nutrients.
This is actually nutrients. Are there also organisms that are somehow linking benthic with pelagic?
But the copepod is in both. They are in the water column.
By the eggs, you mean, in the sediment, or?
They can swim around.
Yeah, but do they feed on the benthic?
They feed on what is in the water column.
Yeah, but not on the benthic.
I don't think they graze.
That's what I mean. They are not so directly coupling it with, compared to organisms that feed directly on the benthos.
Yeah.
And there are also some, no, I think, that feed. I was thinking of some type of fish. No? They feed directly on
fish, yeah.
on amphipods, on benthic organisms. Yeah, then let's move to the biofilm, to the other communities. You mentioned the importance of biofilms in aquatic system. Can you be more specific? Why are they important? I mean, for which specific processes are the biofilms important in, or can be important in certain systems?
So first of all, biofilms are photosynthetic. They
Okay.
produce a lot of the oxygen that we find in coastal areas. And also the biofilm is a nutrient, so many organisms graze and live on the biofilm.
Yeah. And this phototrophic, you say, until which depths of you find these phototrophic biofilms in the Baltic Sea? Until which water depth?
Oh, we have only looked at coastal, so I don't know.
So, and there you have them everywhere, you think, yeah?
What?
You have them everywhere, biofilms with phototrophs in the coastal system?
Very, very deep. There wouldn't be a good thing to be phototroph if there's
That's
no
what I mean.
sunlight.
Yeah, that exactly is my question.
Okay.
But also there are some coastal areas which are deeper, but that's my question. Until, I don't know whether it's known, maybe nobody has investigated that, but until which water depths do you find phototrophic biofilms?
I would speculate that they stop being photosynthetic when there is no sunlight.
Right. And what do you think, which water depths could that be in the Baltic Sea?
It's very dependent, because if like it's clear water, or if it's like more full of algae. Also in the summer we see there's like a really thick layer of sea snot in many places in the heated bay. And I guess no sunlight can go through that.
So light plays a crucial role. I'm wondering that you have not mentioned light. Have you mentioned light in your study, from the biofilm study? You talk a lot about temperature, but what about light?
Yeah, there's of course a change in light, because in the heated bay in the summer, as I said, we see this really thick sea snot. That makes it difficult for us to find our union, because we have to move it all the time and then it floats. And also in the winter, in the control bay, on the other hand, there is a very, very thick ice layer. We cracked it and it was like this thick.
Okay.
So of course the light changed. It would have to go through this layer of ice.
Ah, interesting. So light must be really an important factor, no? for the communities in the two systems. And also when you find differences between the heated bay and the other bay, light might also play a role, right?
Yeah.
Ah, okay, that's interesting. And yeah, you mentioned that there are so many biofilm studies, but mostly have only considered prokaryotes, right?
Exactly.
And relatively few. I have the feeling that in the last years there are some more coming up, looking at also at eukaryotes, but still quite few. But when you talk about micro eukaryotes, which types of organisms do you mean with this? It's not a taxonomic term, micro eukaryotes. So what kind of organisms are that?
This is specified by size. So we removed bigger organisms from our data sets, and we only look at micro eukaryotes. And I don't remember the exact number, but it's under a specific size that this is. And wait a little. No, but
Okay, you remove larger organisms from your...
Yeah, to not interfere the data because we are only interested in the microeukaryotes.
Yeah, I understand that, but my question was, I mean, what are microeukaryotes? I mean, are that single cell organisms mainly
No,
or only?
it's defined by size.
By size,
Mhm.
like 10 or 100 micrometer, or
I don't remember
you
the
don't remember.
number.
Does it also include metazoans?
Metazoans should also be there, and archaea and everything should be there because there's only 4% of the studies that have taken into consideration both eukaryote and prokaryote. So it's really good to look at both domain because also they affect each other. So doing just prokaryote is like doing it half. So in both of the studies in biofilm, we took both domains.
Mm. But well, I first thought that maybe you mean protists, single cell organism protists, protozoa.
Protozoa.
Yeah, mainly protozoa. I guess you cover with your microeukaryotes. Is that right?
Also other organisms.
Also small metazoans.
Yeah, it was everything was there.
Okay, but not large metazoans. Good. But large metazoans, they might also be important sometimes for
Yeah.
biofilms, don't you think, as grazers or so?
Yeah, we were discussing it and we just said we need to standardize. We need to only take the microeukaryotes, but in the future we could make larger studies and take everything.
No, for the sequencing, I understand that fully because you might also get too many sequences from some of the metazones, and then it's difficult to detect the others. But what can you imagine of certain animals, metazones, that are also in your system important for the biofilm?
You mean grazers?
Yeah.
Yeah, actually there is an ongoing study that shows that there is a big difference in the species of what grazers is in the heated bay and what's in the control bay. So that's something we could also use.
Yeah. Is that something like snails or what?
I don't know the exact species names.
Maybe snails and amphipods or so, huh? And could you compare your study to a previously published study on the impact of warming on biofilms? I didn't have time to look for the whole literature, but I found it difficult to find comparable studies. Is there any study? Is there any published study in the literature which also studies the warming on biofilm, the effect of warming on biofilm?
There is a similar...
In the natural system, yeah?
Yeah, there are similar study systems. There is one in Forsmark, but they also have a heated bay. There is actually also, I found a study also in China and England, and around the world they're using similar system but other designs. There was some study they put down concrete and just let biofilm grow, where we used the natural material, the granite stone that is naturally there.
Mm-hmm. And when you sample the biofilm on the stones,
yes,
later on you look at your sequences that you get from that, but do you have any idea how this biofilm looks like? I mean, visually, did you somehow visualize the biofilm? I mean, how it is arranged, how thick it is, which color it has, whether you see a layer of algae or something like that. Did you also make notes, or did you look in a microscope or whatever? I mean, to get an idea how does it look like, the biofilm on the stones?
Well, I was there every time we took biofilm. We didn't note down. We just scraped the biofilm, and sometimes it seemed like the stone is clean, but there is still something there.
Yeah.
So sometimes, especially in the heated bay, it was like you can see it very easily. It's all green matte, as in the picture I showed.
Mm.
And sometimes in the other environment, it's like just sitting there scraping a stone, and it looks like a stone.
Yeah, okay. But you didn't look in the microscope for for what you scraped away from the stones?
No,
you
it
didn't?
was like DNA was extracted and so on. We didn't look before.
Yeah, yeah, and yeah, you probably also realized that there is this biofilm is in a matrix, embedded in a matrix.
It's many organisms. So a biofilm form normally that you put like some kind of hard material. That can also be a ship. Unfortunately, they also do that, biofouling into the water. And the first that will happen is that bacteria will attach, and when the bacteria have attached, other organisms attach on them. So they form this film made of many different organisms. And when the biofilm get too big, too thick, then like they are budding off and in the water column, and then they found a new surface to colonize.